Thank you, everyone. We're at the top of the hour, so we'll reconvene our ACIP meeting today. For the next session, we have Dr. Bill Gruber from Pfizer presenting on the safety, immunogenicity, and efficacy of BNT162b2 in children ages 6 months through 4 years of age. Dr. Gruber, the floor is yours.
Okay, thank you, Dr. Lee. Can you hear me well?
We can, thank you.
Okay, great. On behalf of Pfizer and BioNTech, it is my pleasure to share data supporting the BNT162b2 recommendations in individuals 6 months through 4 years of age. I believe the CDC is advancing my slides for me, so please, next slide. My name is Bill Gruber, and I head the Vaccine Clinical Research and Development Group at Pfizer. Next slide, please. Today's agenda covers the topics shown here with specific attention to coverage of the clinical safety, immunogenicity, and efficacy data, along with the assessment of benefit risk. Next slide, please. I'm now going to share with you the clinical data that supports recommendations for this age group. Next slide. We began in 5 to 11-year-olds before progressing to 64 participants in vulnerable 6-month through 4-year-old age group to achieve the right balance in safety profile and immune response. We found that nearly 19% of 2 to less than 5-year-olds who received 10 micrograms developed fevers after the first and second dose, and 1/3 of these fevers were severe. This could be poorly accepted by parents, reducing adherence to the primary 3-dose series. In contrast, the 3 microgram dose had a much better tolerability profile, combined with comparable to better immune responses to the SARS coronavirus 2 reference strain. Therefore, the 3 microgram dose level was advanced into phase 2/3. To infer efficacy in the pediatric population in the pivotal study, immunological non-inferiority to a 16 to 25-year-old population for whom efficacy was established was assessed in addition to safety to satisfy emergency use authorization immune response criteria and infer efficacy. Although not required for EUA approval, COVID-19 surveillance was conducted, permitting an early evaluation of vaccine efficacy. Next slide, please. Children were administered 2 doses, as shown at the top, 21 days apart, and then a 3rd dose was administered at least 60 days later. Planned follow-up for the study is as shown. Current follow-up includes 1 month post-dose 3 serology and efficacy to the data cutoff of April the 29th. Next slide, please. All safety data that I will present is in the blinded, placebo-controlled follow-up period, first for 2 to less than 4-year-olds, and then the 6 months to less than 2-year-olds. Next slide. Demographics in the 2 to less than 5-year-old age group are balanced between vaccine and placebo groups, whether gender, race, ethnicity, SARS coronavirus 2 exposure, or comorbidities. Next slide, please. Local reactions captured by eDiary over 7 days were mostly mild to moderate in severity, somewhat higher in the vaccine recipients compared to placebo recipients, and did not show an increase from dose 2 to dose 3. Local reactions were higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. There were no grade 4 local reactions. Next slide, please. Systemic symptoms solicited by electronic diary were mostly mild to moderate and comparable after each dose. Fever, fatigue, or other symptom rates were remarkably similar to those seen in placebo recipients at each of the 3 doses, and much lower than those in older age groups immunized with higher dose levels. Fever rates are comparable or lower than other childhood vaccines. Only 3 or less than 0.2% of BNT162b2 participants reported fever greater than 40 degrees centigrade after dose 1 or 2, starting on day 2, day 4, or day 6, with all returning to normal 6 to 7 days after the dose, one of whom had a presentation suggestive of a viral exanthem. None of these required hospitalization, and all resolved quickly. Systemic symptoms were higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. This highly favorable tolerability profile for the Pfizer-BioNTech vaccine should be reassuring to parents and care providers. Next slide, please. In the 2 to less than 5-year-old age group, blinded safety follow-up occurred from the time of dose 2 to dose 3 or cutoff date for a median of 4.0 months, and for dose 3 to the cutoff date for a median of 1.4 months. Next slide, please. Overall, unsolicited adverse events, related adverse events, SAEs, withdrawals were infrequent and comparable between vaccine and placebo groups. 3 subjects, or 0.2%, in the vaccine group withdrew from the study due to adverse events: pyrexia, considered related, status epilepticus, considered unrelated, and urticaria, considered unrelated. 1 participant in the placebo group withdrew due to facial swelling and rash, considered related. There were no deaths reported in this age group. AEs, as defined by system organ class, were comparable between vaccine and placebo groups. Next slide, please. Now, let's look at the 6-month to less than 2-year-olds. The demographics again show balance between the vaccine and placebo groups related to gender, race, or ethnicity, prior SARS coronavirus 2 infection, and comorbidities. Next slide, please. Local reactions in the 6-month to less than 2-year-old group with and without prior SARS coronavirus 2 infection were mild to moderate, with incidence somewhat higher in vaccine recipients. Local reactions were somewhat higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. There were no grade 4 events, and frequency remained relatively the same after each dose, consistent with a very well-tolerated vaccine. Next slide, please. Fever, decreased appetite, drowsiness, and irritability were mostly mild to moderate and similar to placebo rates, lower than those observed in older children and adults, and comparable to those after other childhood vaccines. Fever greater than 40 degrees centigrade was reported by only 3 vaccine recipients, or less than 0.1% for each dose, starting on day 1, day 2, or day 3, with all returning to normal 5 to 6 days after the dose, 2 of whom had a concurrent viral infection. 1 fever greater than 40 degrees centigrade was reported by a placebo recipient after the first dose. None of these required hospitalization, and all resolved quickly. Systemic symptoms were somewhat higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. Again, the overall incidence and low severity of systemic symptoms speaks to a favorable tolerability profile that should be reassuring to parents and care providers. Next slide, please. Safety follow-up included blinded follow-up from the time of dose 2 to dose 3, or the cutoff date for a median of 6.3 months, and from dose 3 to the cutoff date for a median of 1.3 months. Next slide, please. Any adverse events, related adverse events, SAEs, withdrawals were infrequent and comparable between vaccine and placebo groups. 3 participants, 0.3% in the BNT162b2 group, withdrew from the study due to adverse events, all related. 2 due to fever greater than 40 degrees Celsius, 1 of which had a viral exanthem that was unrelated. 1 participant withdrew to a generalized rash on the face and trunk. There were no deaths reported in this age group. AEs, as defined by system organ class, were comparable between vaccine and placebo recipients. Next slide, please. Few adverse events of special interest were recorded. Predominant categories were potential angioedema and hypersensitivity, comprising mainly urticarias and rashes, balanced between groups. No CDC-defined adverse events of special interest were observed. Next slide, please. The carefully selected dose level of 3 micrograms for the BNT162b2 vaccine was shown to have a favorable safety profile and was well tolerated in infants, toddlers, and very young children. Vaccine reactions were mostly mild to moderate and short-lived, with systemic reactions comparable to placebo. Reactions were comparable after dose 1, 2, and 3. The unsolicited adverse event profile mostly reflected reactogenicity or common childhood illness. Favorable safety profile should encourage vaccine adherence for each of the 3 doses. Next slide, please. Demonstration of non-inferior immune response in children less than 5 years of age compared to immune response in 16 to 25-year-olds after 2 doses was judged by the FDA as sufficient to meet immunologic success criteria for emergency use authorization. Next slide, please. Immunobridging criteria for 2 to less than 5 years of age were not met for geometric mean ratio, but were met for seroresponse after dose 2. Geometric mean neutralization titers are shown for 2 to less than 5-year-olds, light blue, compared to those in 16 to 25-year-olds, dark blue, after dose 2 at the top of the slide. I draw your attention to the GMR in the tan box at the top of 0.61, which failed to meet the lower confidence bound of 0.67. However, the difference in seroresponse in these groups shown at the bottom right of minus 0.9% and 95% lower bound of minus 4.3% are above the minus 10% criteria. Success was not declared due to not meeting the GMR criterion. Next slide, please. However, in consultation with the FDA, a similar comparison was made between GMTs in the 2 to less than 5-year-olds to greater than or equal to 65-year-olds in the pivotal efficacy trial. In this case, the GMR was 1.92 with a lower bound of 1.56, well above the 0.67 non-inferiority criterion. Although this was a post hoc analysis, vaccine efficacy in the greater than or equal to 65-year-olds was 94.5%. So a non-inferior immune response seen in the 2 to less than 5-year-olds is likely to predict efficacy. As you will see, 35.9% efficacy in this age group was observed in 2 to less than 5-year-olds after the second dose. Despite the likely benefit of a level of 35.9% efficacy after dose 2, based on the growing importance of Omicron, we judged a 3-dose series as important to provide improved protection, just as is required for every other age group. Next slide, please. Immunobridging criteria of 6 months to less than 2-year-olds compared to 16 to 25 years of age were both met. Immunobridging criteria were met for both the geometric mean ratio, shown 1.03, lower bound of 0.9, above 0.67, and difference in seroresponse at 1.7%, well above the minus 10% criterion. But again, based on observed efficacy data I will share shortly, we judged that just as for every other age group, in an Omicron predominant environment, a 3-dose series would prove necessary for a high level of protection. Next slide, please. Immunobridging criteria in the 2 to less than 5-year-olds were met for both GMR and seroresponse, comparing post-dose 3 responses to post-dose 2 responses in 16 to 25-year-olds, which infers vaccine effectiveness after dose 3 in this age group. The median dosing time between dose 2 and dose 3 was 10.7 weeks. The geometric mean ratio observed was 1.30, lower bound of 1.13, well above the 0.67 success criteria. Likewise, difference in seroresponse was 1.2%, lower bound of minus 1.5%, well above the minus 10% non-inferiority success criteria. Next slide, please. Now, let's look at the 6 month to less than 2-year-olds. Immunobridging criteria were met for both GMR and seroresponse, again inferring vaccine effectiveness in this age group. The median dosing time between dose 2 and dose 3 was 12.9 weeks. GMR was 1.19 with a lower bound of 1, well above the 0.67 required success criterion. Difference in seroresponse was 1.2% with a lower bound of minus 3.4, again well above the minus 10% required success criterion. So for both younger children as well as the older children, immunobridging criteria were met, inferring vaccine effectiveness. Next slide, please. Given that most current COVID-19 cases are caused by Omicron, we also evaluate the ability of 2 and 3 dose immune sera from 6 months to less than 5-year-olds to neutralize Omicron compared to sera from adults. In this graph, the older comparator group is a 24 to 74-year-old adult sentinel cohort from the licensure trial. Immune responses to Omicron, shown in pink, were compared to those of the reference vaccine strain, shown in blue using a plaque reduction neutralization assay. GMTs are shown on the y-axis and age groups are shown left to right. As you can see, we see comparable immune responses across all three age groups to the reference strain and Omicron. However, as reported by several groups, Omicron responses after only 2 doses are low and uniformly so across the pediatric and adult age groups. Low Omicron neutralization titers after 2 doses seem to correlate with lower efficacy against 2 vaccine doses for Omicron. Next slide, please. However, this picture changes when serum samples after 3 doses are being evaluated. In this data set, an adult comparator group was used that received the 3rd dose at a similar time interval after the 2nd dose as the pediatric group at about 11 to 13 weeks. Neutralizing antibody responses were measured using a fluorescent focused neutralization assay. Left to right, for 6 months to less than 2-year-olds and 2 to less than 5-year-olds, the Omicron-specific neutralization titers after 3 doses are far higher than those on the prior slide after 2 doses, and the Omicron-specific titers are very similar across age groups. Most importantly, the pediatric group titers are essentially the same as in the adult group, predicting that similar efficacy as shown in adults could likely be observed for the 6 months to less than 5-year-old age group after dose 3. I will shortly share with you descriptive data showing observed post-dose 3 efficacy matching this prediction. Next slide, please. So here are the immunogenicity conclusion in 6 months to less than 5-year-olds. Immunobridging criteria were partially met after dose 2. All immunobridging criteria post-dose 3 in young children required for an emergency use authorization were met for both age groups, inferring effectiveness. Omicron neutralization titers were low after 2 doses for pediatric and adult cohorts, but increased substantially after a 3rd dose in the 6 months to less than 5-year-olds to similar levels observed in adults. Thus, as has been observed in other populations, a 3rd dose is required also for the 6 months to less than 5-year-old age group to ensure a more robust protection against COVID-19 due to Omicron than can be achieved after 2 doses, and despite the fact that we saw some level of efficacy after a 2nd dose. Next slide, please. So what have we learned from the adult and pediatric experience about the potential vaccine efficacy against COVID-19? Next slide, please. Here's a summary of descriptive observed efficacy in the blinded follow-up period. Efficacy against the Delta variant is shown in blue, and efficacy against the Omicron variant is shown in pink. The Delta surge ended prior to the 3rd dose, so no children were exposed to Delta after the 3rd dose, marked as NA. Observed efficacy against Delta post-dose 2 is favorable across all age groups post-dose 2. The emergence of Omicron presented a new challenge. Shown in pink on the left, you can see that for children 6 months to less than 5 years of age post-dose 2, efficacy was 21.8% in the 6 months to less than 5-year-old age group, and correspondingly as shown for the other subgroups. This is consistent with the poor antibody response after the 2nd dose. Now look what happens after the 3rd dose. Cases post-dose 3 in this clinical study were confirmed to be Omicron. Efficacy rises to 80.3% overall and correspondingly high for the respective age groups. This is consistent with the higher antibody response seen after the 3rd dose against Omicron, like that of older children and adults, and consistent with corresponding higher efficacy in older children and adults after a 3rd dose against Omicron. Next slide, please. Overall, vaccine efficacy observed over the period of blinded observation after dose 2 is shown here. Looking at the 2 to less than 5-year-old age group in the middle, 35.9% and 34.3% benefit for a valuable population was shown. This indicates likely benefit after the 2nd dose for at least children 2 to less than 5 years of age. Next slide, please. Note cases in placebo recipients in red and in in red and vaccine recipients in blue on this plot. For 2 to less than 5-year-olds, there appears to be increasing efficacy over 210 days, and the interval is important, of observation after dose 1, largely driven by the efficacy after the second dose, since cases after the third dose represent a small minority of total cases. This supports a likely benefit after the second dose in this age group. Next slide, please. The potential benefit of the second dose is not as readily apparent in 6 months to less than 2-year-olds, but perhaps there is some separation of the curve at some distance from the second dose, shown on the right, suggesting the possibility of maturation of response and protection. The goal of the third dose is to shift that widening of the curve to the left. Next slide, please. Some vaccine effectiveness after dose 2 is likely to be inferred by the observed 35% efficacy in at least 2 to less than 5-year-olds. However, the immune response data shared with you today, descriptive efficacy shown on the right, and the experience in older children and adults supports the following. 3 doses of the BNT162b2 vaccine are in the best interest of children to provide a high level of protection against Omicron, observed to be 80.3% with lower bound of 95% confidence interval of 13.9 in 6 months to less than 5-year-olds with point estimates in smaller subgroups as shown. So next slide, please. So what can we conclude about efficacy? As demonstrated in other pediatric and adult age groups, 2 doses of BNT162b2 are protective against variants of concern such as Delta, but do not provide adequate protection against Omicron. As demonstrated in other pediatric and adult age groups, a third dose is necessary to provide high protection against Omicron. Next slide, please. Ongoing pharmacoepidemiology, including 5 pediatric studies, as well as proactive risk mitigation and pharmacovigilance, will be extended to this younger age group. Next slide, please. The potential benefits of vaccinating children 6 months to less than 5 years of age outweigh the known potential risks. This age group of 6 months to less than 5 years of age is currently unprotected. Protection against COVID-19 is critical, particularly in light of the unpredictability of potential new waves and emergence of new variants of concern. Available safety, immunogenicity, and efficacy information support a highly favorable benefit-risk profile for administration of 3 doses of BNT162b2 at 3 micrograms to children less than 5 years of age. Next slide, please. Pfizer and BioNTech wish to thank all those listed who made this investigation possible, and I and my Pfizer colleagues will now be happy to entertain questions. Thank you.
This presentation is open for questions. Dr. Long.
Hi, thanks, Dr. Gruber. Can you go back to slide 30? And I'm sure you said this early in the presentation, but I just want to be sure I understand showing this efficacy after the third dose. What is the definition of the clinical problem that you're protecting against? Is it infection? Is it
so it's symptomatic illness with at least one symptom.
One symptom. So it's the CDC definition. Okay. And then, yes, go ahead.
No, I was just saying it's close to that. That's correct.
Okay. And then I didn't write down the number, but the slide that shows it's the Kaplan-Meier plot of cumulative something over time in older people after 3,
That's
I think.
probably slide 32 in the 2 to less than 5-year-olds. Maybe
yes, I think it was very close. Yes. So, and this was an assumption slide trying to show that there was efficacy from a third dose, that these people likely received a third dose.
Yeah, so I think, let me see if I can explain this slide in a little bit more detail. So
yes.
In the interest of time for the overall presentation, I didn't get a chance to really describe all the details here. But what you see on the x-axis is the timing from dose 1. So this includes, of course, children who have had now dose 2, and you can anticipate if I put a line there, it'd be at about day 21 for dose 2.
Okay.
So you can see, and then dose 3 was basically, as I told you, about 11 weeks or so out from the time of dose 2. So you can, and remembering that the total number of cases that we had in this age group post-dose 3 were 7. So the fact is that most of the cases that you're seeing here are actually still post-dose 2. And that, I think, that what drives the conclusion and the estimate of around 35% or efficacy or so is the fact that most of the efficacy you're seeing here is driven post-dose 2. And then, of course, I share with you the table that speaks to the post-dose 3 efficacy, which is specifically focused on looking after dose 3, if that helps.
Yes, I was really pretty interested in time since any dose. So it looks like you would just estimate that there was 100 days, at least 3 months after the last immunization that anybody could get here. And there was still was
I think it was encouraging to us, and again, this could be well due to some maturation of immune response. Obviously, there are contending forces at play. We know that essentially everybody, particularly after 2 doses, shows waning protection against symptomatic disease, less so against severe disease, and that protection is more sustained after a 3rd dose. But this would suggest, if you look, and again, I don't want to make too much of the curve because the numbers start becoming smaller, farther out you go, but you do get some impression that maybe there's even some widening here. And I think it's important, and I think you'll see this in the CDC slides that we've had a chance to take a look at, that it's a little dangerous to compare efficacy against, across, different studies with different follow-up. But I do think the follow-up here out to 210 days is particularly important because it does suggest that, at least in this age group, there may be some persistence of protection. And of course, our goal is not to have children wait out to 210 days. Our data would support giving that 3rd dose so we can boost that efficacy up to 80% or more.
Thank you. And the very last thing is, I assume that this is by definition of 1 symptom and positive PCR.
Yes. These are nucleic acid amplification tests, either based on a central lab test or an approved test from a local laboratory, but it had to, we had to meet specific predefined requirements for that to be acceptable. So this was not home testing. This was all laboratory testing.
Thank you.
Thank you, Dr. Paling.
Okay, I have 2 questions. First is, could you remind us what the placebo is?
Paling.
Perfect. And then the second thing is, how much difference in timing between the 1st and 2nd dose did you have? Did they all cluster right at 21 weeks, or do you have some experience about spreading the doses between dose 1 and 2?
So I think in this particular study, I don't know, we have a slide that represents, but I think like all our other studies, it's very, very highly clustered between the 1st and 2nd dose at around 21 days. And then, as I explained, after the 3rd dose, the range is close to about 11 to 13 weeks, 2nd to 3rd dose.
And we do have a slide, Dr. Gruber, it's Charu, if we want to pull up slide 62, if that would help.
I'll leave it to Dr. Paling. Do you want us to pull up the slide?
Thank you.
Yeah, all right, so let's see if it's slide 62 if we want to pull that up.
We see it, thank you.
This is the dosing again between dose 2 and dose 3. I think Dr. Paling was asking between dose 1 and dose 2. And again, I think 90-something percent of my recollection, as with all our other studies, is in the between dose 1 and dose 2 occurred within the 21-day period.
It was 99%. Thanks.
99%. There you go, even higher.
Thank you. Ms. Bata?
Thank you, Dr. Kruger, for this brief summary. I appreciate it. Just in looking at the 6 months to under 5, slide number 34, it looks like there's there are very wide confidence intervals, and in each of the subgroups, they're below, they cross 0. These were cases that were observed after February 7th until what point in time?
Up until the data cutoff. And so there are actually 2 lines of evidence that indicate that these are all Omicron cases. One is the timing, so they were all after actually February 7th, but also based on sequencing. They're confirmed to be Omicron. Let me just comment about the confidence intervals. We recognize, obviously, the 6 months to less than 2-year-olds, you're talking about 3 cases here. And I think the argument we're making is that overall, when you roll this up, the findings are consistent with what we see in other populations. I think it's important also to keep in mind that, we think about our other age groups, 5 to 11, 12 to 17, for which we provided in the past, or I'm sorry, 12 to 15, for which we provided data in the past. The total numbers of subjects were not much bigger than what we have here. And what we, didn't do at that time, nor were we required to do, was sort of break it down by smaller intervals. So when you talk about the 6 months to less than 2-year-olds, that's obviously a smaller proportion of the population. There are fewer total children that were sort of recruited there. And you can see the denominators as shown there. In a smaller age segment, who may or may not have, equivalent exposure, perhaps are in a more protected state. So again, we actually look back. I don't think we have the slide in this set, but we actually look back. If we were asked, for instance, to look, in 2-year intervals between the 5 to 11-year-olds, we would have found some for which the lower confidence interval was below 0. And yet it was the nature of the overall span of 5 to 11s that provided enough cases to be, confident that in that age group, given the immune response as being comparable to what was seen in other populations, that they would likely be protected. So, I'm not dismissing by any means that, okay, the number are fairly small, but I would put this in the context for the 6 months to less than 2-year-olds of what you've seen in other populations, the immune response that matches up in terms of non-inferiority, and the high efficacy that's seen in other populations in this range where that type of antibody response has been achieved.
And I really appreciate the fact that these numbers are small. Are you planning to continue to assess the vaccine efficacy as time plots out? Because I think you'll get bigger numbers and more certain point estimates.
Yeah. So it's a very important and great question. There are 2 ways, obviously, we're going to do this. We have the capacity, obviously, up until the point that we start rolling individuals over who are all getting vaccine, which we'll have some obligation to do within the clinical trial. And in fact, already are doing this at 6 months after individuals have been randomized. But we will have additional cases under a blinded, placebo-controlled period that should give us more precision on these numbers. And of course, after licensure, just as we always do, we'll be taking advantage of the Kaiser Permanente population that served us so well in the past to better identify true effectiveness. And of course, it will give us the long-term benefits as well of durability, of protection, not only against milder disease, but severe disease. And I might add, I think there's great comfort that we have had that it does appear to be the case in older populations that protection against severe disease leading to significant medical attention or particularly hospitalization seems to be better preserved. And given that the antibody responses are well matched here and the vaccine efficacy that we're seeing is already high after a 3rd dose, it gives me real confidence that we should see the same thing in this population, but we'll definitely study it.
Thank you.
Thank you. Dr. Sanchez.
Thank you, and thank you, Dr. Gruber. And I'm going to be asking similar lie?
Yeah, so there were instructions given to the investigators, consonant with other instructions for pediatric vaccines, that children less than 2-year-olds would have it administered in the thigh, and children over 2 years of age were to have it administered in the arm.
And along with that, when you reported the swelling as an adverse effect, was that local swelling at the site, or were you talking more about adenopathy and in the older children, inguinal swelling and adenopathy?
Yeah, this was local swelling. I will say, and we actually have a slide with this that was presented at the VRPAC, where we talked about the system organ class comparisons, that lymphadenopathy was extremely infrequent, less than we'd seen in other populations in both age groups. So, from that perspective, it seems that we're seeing less of that manifestation of vaccination.
And how about the prematurity issue? Were any of these?
This is the advantage of having had a chance to listen to Moderna's talk. So we looked that up real quick, and 1.6%. I don't know that we have immune response data. I think we also are obviously looking as to what the future course will be to investigate vaccine in children less than 6 months of age, where I think we'll have a better opportunity to identify potential implications of prematurity as we get into that younger age group. And we're in discussions with FDA about how best to do that.
And how about concomitant vaccines? I'm sorry, Dr. Lee, I hope this is okay.
No, please go ahead, Matt.
Yeah. So concomitant vaccine use, again, was a temporary exclusion for inactivated vaccines and rotavirus 2 weeks before or 2 weeks after. For other live virus vaccines, it was 4 weeks. And so we don't have concomitant vaccine data when the vaccines were administered at the same time. Obviously, that becomes that much more important within the crowded vaccine schedule, for which you have more limitations as we work to look at children less than 6 months of age, and we're clearly going to have to think about that there.
And then finally, it doesn't seem, and just correct me, that you did not do any surveillance for positive PCR, so we don't know about asymptomatic infection or transmission.
Yeah, we did not do routine surveillance in this population to look at impacts on transmission. I think that's going to, await real-world effectiveness studies, which I think have been useful in identifying potential for protection against transmission.
Thank you.
Thank you, Dr. Bell.
Thank you, and thank you very much for the presentation. I also appreciate the efforts to evaluate efficacy, which I know is difficult, obviously, in this situation. And with that in mind, it makes the immunologic data that much more important. We're really relying on immunobridging. And so I got a little bit confused as you were showing the immune response with the comparison populations. It seemed like sometimes it was adolescents, sometimes it was young adults, sometimes it was people older than 65. Then there were other age categories. And I just wondered, as I say, I got a bit confused, and I just wondered if you could help us, if you could explain a little bit more for us what the comparison groups were in the immunobridging studies.
Yeah, so I'm happy to do that. So for the purposes of meeting emergency use authorization criteria, the population chosen to which we compared was 16 to 25-year-olds from the pivotal study. Now, why was that the case? Well, as is often the case, you want to make your comparison to a population for which you have demonstrated efficacy in a well-controlled trial. And so 16 to 25-year-olds are from that population. Now, why 16 to 25? Well, that actually provides the most rigorous comparison, because we know that the younger you were in that age group, from 16 on up to above 65, the higher the antibody response would be. So it sets the bar pretty high to be able to demonstrate non-inferiority. So having demonstrated that, you could be confident, okay, I've demonstrated it, against a pretty rigorous assessment. And therefore, I should be confident that we should see a level of efficacy, at least that of which was seen in the efficacy trial for individuals that were enrolled in that trial. So that was the basis for the non-inferiority criteria, and that was true whether we were looking at 2 doses in the pediatric population under study or 3 doses. We were comparing to 2 doses in the 16 to 25-year-olds because that translated directly to the pivotal efficacy trial, and it was agreed upon by the FDA as then inferring effectiveness. Then in addition to that, to further inform our thinking about, okay, but the world has changed. The world has changed where we're now encountering Omicron and its descendants as the major threat. And we all have now seen that 2 doses of the vaccine just don't work as well against Omicron. And that was, again, affecting our thinking, in the context of thinking about what the future should be. We wanted a vaccine that could protect well against Omicron, not just at a 35% level, but at a high level, 40%. 30% offers some benefit, but the goal here is to try to prevent Omicron as best one can. So in that context, we then did comparisons of post-dose 3 to post-dose 3, the younger population to the older population, and showed that post-dose 3, we were matching. If you go, actually, we can go to slide 27, because I think this is a fundamental slide to understand this. So you can see here that the goal was really after dose 3 to show, can we achieve antibody levels comparable to an adult population for which we know we've been able to demonstrate efficacy, and for which 3 doses of vaccine from real-world effectiveness data is showing efficacy that's relatively high in the 70–80+% range. And so you can see that given the limitations of the sample size, you have overlapping confidence intervals. You see 139 for the 6 month and less than 2-year-olds, 113 for the 2 to less than 5, and 155 for the 18 to 50-year-olds. So this, combined with the descriptive efficacy, says this is on the right track. We have every confidence that we're going to see high efficacy against Omicron. Hopefully that explains.
Thank you. Dr. Lee, could I just ask a follow-up question, please?
Please do.
Could we go to slide 22, please? So this was what started my confusion. And just to ask you a little bit more, what the thinking was about using a greater than 65-year-old comparison population. I recognize this is only after dose 2, and it's a small slice of the overall picture. But nonetheless, I just got a bit confused. Thank you. Yeah.
So this was, again, I want to emphasize, this was a post hoc analysis. So back in February, when we were first greeted with the information that I've shared with you, and Omicron was really taking hold, and we were trying to assess, okay, what is in the best interest of children? We said, well, look, the individuals over than 65 years of age, it isn't as if they had no efficacy. They had quite a high degree of efficacy. So arguably, could one have chosen this as a comparison group of non-inferiority? And how would we have fared in that circumstance? And so we did that in consultation with the FDA and demonstrated that, yes, if we made that comparison, not only would we have passed, you can see that the lower confidence interval suggests that we're well above. At 1.56 geometric mean ratio, if you see there on the right-hand side, we're well above the criteria of 0.67 that would be required if we made that comparison. So if the goal was really to compare to a population with efficacy, we would have passed at that point, suggesting, okay, there probably is some likelihood of benefit. And in fact, for the 2 to less than 5-year-olds, looking back as we just shared with Dr. Long and showed during the core presentation, you can see efficacy in that age group. So this was just another measure to give us some sense of where we were. But I want to emphasize that we considered that 35% or slightly higher efficacy was just not enough for us to be confident that we were doing right by children, and why we needed a 3rd dose. 2 doses could offer some potential benefit, but the goal here is to get to a high level of efficacy and get children vaccinated as quickly as we can.
Thank you. Dr. Daly, do you have a follow-up question to this? Because I actually wanted to ask a clarifying question on this thread.
Why don't you jump in, Dr. Lee?
Okay, thank you. If we could go to slide, I just want to confirm that I understand the data. I'm actually going to ask you to run through a couple of slides for me because I'm also confused. So on slide 22, oh, that's where we were. If you go to slide 21, so this is post-dose 2, the neutralization NT50 titers, post-dose 2 in the original population that was per protocol. Is that right?
Right, that's correct.
Okay. And then the sort of post-hoc analysis is what you showed, the comparison to the older adults, which we understand that they probably did not have as great immunogenicity on slide 22. Oh my gosh, 22, sorry.
22, right.
Okay. And then in slide 23, I think what you're saying is that when you did that comparison, you didn't see that low GMR as you did for the younger kids. So it looked equivalent following 2 doses for each.
Well, yeah, so let me be clear about this slide to make sure we're all talking about the same thing. So this is in the 6 months to less than 2-year-olds.
Yes.
So back in February, when we had this data, we had met the non-inferiority criteria. And ostensibly could have proceeded to potentially say, okay, we met non-inferiority in this age group. Based on that inferring efficacy, we could proceed. But again, the conscious decision was made based on the efficacy that we were seeing in Omicron in both age groups, that it was in the best interest of children to wait until we had the 3rd dose data and we could show, convincing high level of efficacy.
Okay.
And that was, and we had to wait for that 3rd dose. Children were just starting to get dosed in January with that 3rd dose.
Okay, thank you. Yes, I got the age groups confused. So the younger groups were okay, at least they seemed equivalent post-dose 2. The older groups looked lower compared to the original population.
I mean, if you don't mind, if we go back to 21, because I just want to clarify, again, it wasn't, we passed one test, the seroresponse test, but we didn't pass the geometric mean ratio. And so, the criteria was we really needed to pass both.
Yes.
But there was some suggestion. And again, I think given, let's face it, given what we've now seen in terms of efficacy, one can argue that this is not as precise a measure of what you're likely to see. And hence the reason why it made some sense to say, well, look, there's some evidence here that based on seroresponse, we might have efficacy. Let's look at the older population as a comparator and see if that, would have been a better predictor. And in fact, in retrospect, it probably is, in this context.
Okay, thank you. And then if you go to slide 24, then this is post-dose 3 of the 2 to less than 5 compared to post-dose 2 of the 16 to 25. So it, in a way, it looks a little higher, but essentially equivalent to. So I'm just trying to understand. So I'm thinking my interpretation of this is that 3 doses is as good as 2 doses in the 16 to 25-year-old population here.
Yeah, well, statistically speaking, you can see that the lower bound is actually above 1. So you could argue that the geometric mean titer is, statistically actually higher than it is in the 16 to 25-year-olds after a 3rd dose in the 2 to less than 5-year-olds compared to, after a 2nd dose in the 16 to 25. So it certainly meets the non-inferiority criteria and is actually a little bit greater. And if you don't mind, we can go and just, speak to the same thing on slide 25, which is the 6 months to less than 2-year-olds. You see the same thing, that we're right at, with the lower bound of 1. The FDA had hoped that at least the point estimate would be 1. Well, we've got the lower bound of 1. I mean, to meet the criteria, we needed to be above 0.67. The FDA said, appropriately to try to, increase the likelihood that protection would be seen, that the point estimate should be 1, but in fact, our lower bound is 1. So in both instances, we actually exceeded the criteria that was required for this comparison, and hence the reason, as you've heard today, this is part of what supported the approval for emergency use.
Okay. So I think, thank you. So basically, so the 2 doses in the younger age groups, I'm going to speak very broadly. I recognize it's really for the 2 to 5-year-olds, 2 to 4-year-olds, is lower. The immunogenicity or the GMR doesn't meet it for following dose 2, but it clearly meets it following dose 3. So that's really helpful to understand. I'm just going to say that out loud to make sure that that is correct. That is my interpretation. Does that seem right, Dr. Gruber?
Yeah. And in fact, I mean, that's, if we go, again, just to, maybe we can highlight it in the conclusion slide, which I think was 28 for the immunogenicity. If you wouldn't mind going to that slide. So immunobridging criteria were partially met after dose 2. And I'd say along with that, that conferred, at least in the 2 to less than 5-year-olds, some degree of efficacy that we in fact did see. Arguably, there's very little to see in the 6-month 2-year-olds, except potentially out to, distal from vaccination, which again could represent immune response. But I don't want to make too much of that given the small numbers. But clearly, after the 3rd dose, we're meeting the immunogenicity criteria. And then when you saw the descriptive efficacy, that's consistent with the older adult populations and older children.
Okay. And then to your point about efficacy being the thing that we'd actually like to achieve, the numbers are small, so the confidence intervals are quite wide in general. And then for the subgroup analysis, go below 0 because the numbers are very small.
Yeah, again, as I think with all the data that's being presented today, it's hard to have really high confidence intervals because I think everybody's struggling with getting enough cases in time. And remember that the efficacy was not a requirement for approval, right? The efficacy, because we can't predict when the epidemics will occur. I mean, as you saw, after the 3rd dose, we had no Delta, right? So there was no way to sort of look at that. But I think, looking, if you would, at slide 34, to your point, just so people have this in front of them as we're talking about it, for the group for which we at least have numbers to have some confidence and anticipating that given the antibody response looked good in both the 6-month to less than 2-year-olds and the 2 to less than 5-year-olds, I would contend that the 6-month to less than 5-year-old efficacy, where the lower bound is above 0 at 13.9%, is likely to be a close reflection of the truth. Obviously, as we heard, there's interest in capturing more cases. We'll do that over time. But I don't think that this is a circumstance where people should bet against the vaccine providing protection in 6-month to less than 2-year-olds that's higher than after 2 doses. I think that is, in fact, what we'll see.
Okay, thank you. So I'm just going to hit my 2 high points and then I'm going to turn it over to the others. But my two sort of high level comments are that I recognize that we can't actually compare neutralizing antibody titers across the different vaccine products. And that is my recollection from early in the pandemic. But it seems like it is challenging now as we're starting to leverage and use that as the key to get to authorization or approval, particularly for the younger age group. So comparability across trials, I feel like seems more important now than ever. And then this is just a generic comment. And then the second thing I just wanted to state is that, it's been really hard for us as parents and as pediatricians because recognizing that older adults were the group that was dying at the highest rate early on in the pandemic, it does feel like children have become the lowest priority in the pandemic. My request is just to continue to make them a priority. I think the durability of both effectiveness, immunogenicity, and efficacy are incredibly important. So just would really strongly ask that we continue to take a look at, longer intervals, how many doses are appropriate, et cetera, as we have this population in front of us that is still highly unvaccinated. So I still think rather than waiting for the post-market effectiveness studies to come through and for us to then retrospectively look at that data, I do feel like this is an opportunity for us to prioritize kids, pregnant women, immunocompromised populations, and make sure we can do deeper dives into that prospectively, given that we continue to have a lot of uncertainty around the data. So thank you for indulging.
Dr. Lee, can I respond to both of those 2 things?
Oh, sure.
Okay. All right. So the last one, I couldn't agree more. After all, I'm trained as a pediatrician and infectious disease and vaccine specialist. So, my heart is in making sure that this happens. And I think, as you may have heard in the VRBPAC presentation, we have plans to do just the things that you're talking about, not solely relying on the real world effectiveness, but also working to look at intervals, particularly as we get out of a pandemic period. I think as far as the former is concerned, you're absolutely right. It's dangerous, and I think a false way to go to compare efficacy against trials. And certainly in the circumstance where interval follow-up is particularly important, and we know that there are differences that occur over that period of time. Likewise, for immune response, it's dangerous and inappropriate to compare antibody responses, particularly given the differences in the assays. Where you can, though, make those comparisons, as I've tried to point out here, though, in a well-controlled circumstance where you're essentially running, in this case, the pediatric assays at the same time as you're taking sera from the adults. So these are not historical results. We're running these at the same time in the same assay to make these comparisons. And where you can show that, hey, we've got good antibody response after the third dose compared to what we see in adults for Omicron, I think you can take that to the bank with some confidence that, okay, that's likely to predict efficacy, as it did here, given the descriptive efficacy we provided.
Thank you. Dr. Daly.
Yeah, thanks so much. Some of the issues that I was going to raise were covered by Dr. Lee, but I do think there's still a couple points that I would like to discuss. I mean, I think I'm going to try to focus my comments on the entire age range of 6 months to less than 5 years. What I come away with is that the FDA established immunobridging criteria for emergency use authorization, and those criteria were not met for a 2-dose series, and they were met for a 3-dose series. And so I think of this as a 3-dose primary series. And I think that's important because I don't want parents to get the impression that 2 doses is sort of good enough. And so, I think when we talk about whether there's some separation after 2 doses, I think we need to be cautious about that. I'm interested in seeing that data, but again, I think we all need to approach this as we have evidence that this met criteria established by the FDA for a 3-dose primary series. Then I think this is a good slide to stay on if we could, which is that I also feel like the message that I've heard from parents is they want to know the full story, and it's in the interest of transparency, and then they want to make the best decision that they can. So if parents are told we think a 3-dose series is 80% effective or has a vaccine efficacy of 80%, I'm concerned that parents are going to misunderstand our certainty in that estimate. So could you talk to the audience about what a confidence interval of 13.9 to 96.7 means when parents are trying to interpret a statement or a press release that we have vaccine efficacy of 80%? And then I have some additional questions.
Yeah, so let me address the second question, I guess, first. I mean, the first question. Well, actually, let me speak to the first question. Obviously, we are getting approval as a 3-dose series, but I think it was important to share the post-dose 2 data because I think there have been some misconceptions that nothing was being seen in terms of the potential for at least some efficacy after the second dose, and it was really a conscious choice on our part to move forward with a 3rd dose because we thought, again, to your point, it's in the best interest of children. And I really think that there is no circumstance in which, frankly, 3 doses aren't going to be required to provide that level of protection. As far as the confidence interval is concerned, obviously over time, there's expectation that we will get more precision. And as I mentioned, we have the potential to acquire additional cases since the cutoff of April the 29th, which of course still was during a period of time when Omicron was circulating, that we can further inform what this efficacy is. But we're at this point in time now, and this is the data we have, and I think the need is urgent to start immunizing children to get 3 doses in because the future is uncertain, and we often find ourselves on our back foot, trying to catch up. I think in this context, it really means that 95% of the time, looking at the top number here of 80.3, that the efficacy is going to be somewhere between 13.9 and 96.7, but with the center of the curve around 80.3%. So most of the curve is in a circumstance where you're likely to have a high degree of efficacy given this case split, remembering that this is a 2-to-1 randomization. I don't think that this is much different than other sorts of efficacy that we've been hearing about today, whether we're talking about 2 doses or 3 doses. In some circumstances, I think the confidence intervals, depending on the definition, can be in this range. So my sense is that the totality of the data needs to be kept in mind here, that the nature of what we see in terms of antibody response, particularly to Omicron after a 3rd dose, provides some reassurance. The experience that we've seen in other populations after a 3rd dose where that level of antibody has been achieved provides us some confidence. And limited as these numbers may be, they're in the right direction. They're consistent with what we're seeing, and our anticipation is that real-world effectiveness data, as well as any further data that we can acquire from the controlled trial, should also be consistent.
Yeah, thanks for that, Dr. Gruber. I just think that if there's a press release about 80% vaccine efficacy, that there's some assumption that there's a fairly high degree of certainty around that, and this shows that the confidence intervals are wide. And again, I think, I have the same hope that you do, that the efficacy is high and matches the immunogenicity data. But I feel like, in the interest of sort of full transparency to parents, it's to me appropriate to acknowledge the uncertainty around that as well. So if you could go to slide 20.
You are muted. You can mute or unmute yourself by pressing *6.
Series, and I'll wait for a sec till we get to slide 27. I'm still seeing slide 34.
There we go.
Yeah, so part of my confusion stems from the number of different comparisons were made and then who the comparison group was. So a 2-dose series in kids 6 months to less than 5 years of age did not meet the criteria for emergency use authorization, but a 2-dose series in adults did. And so when we're comparing, is this comparing a 3-dose series in kids to a 3-dose,
yes.
three vaccines received, but in those 18 to 50-year-olds, that's a different context, correct? Because that's 2 doses, which met criteria, and then a subsequent booster dose, which I think of as different than receipt of a 3-dose primary series in kids. And the reason I bring this up is also in the it's both, how do I interpret this? And then it's also
let me see if I can. So just to be clear, that the comparison here is different than what we were talking about in terms of the non-inferiority criteria, non-inferiority comparison to meet success criteria. In this case, we're specifically focused on, okay, what is the coin of the realm here in terms of something we could potentially hang our hat on that would allow us to predict efficacy? And I think the level of antibody response that we've seen to Omicron after a 3rd dose in older populations, it comes the closest to anything that we have to sort of saying, okay, when we got a higher antibody response, we had higher efficacy. And so making that assumption in this context where these individuals got 2 doses, but note the interval with a booster dose at basically 13 weeks, which is pretty close to the 10.6 weeks and the 12.9 weeks that you see in the 2 to less than 5-year-old and 6 months to less than 2-year-olds, respectively, after they got the 3rd dose. So in my mind, whether we call it a primary series or we call it 2 plus a boost, in this circumstance, we're comparing the same type of series. And I think you've heard argued, I would subscribe to this, that in a world of Omicron, we're frankly talking about a 3-dose series for everybody. 3 doses are required to protect against Omicron. And so we've done the best job that we could to compare like to like 3 doses with the 3rd dose, with the difference between the 2nd dose and the 3rd dose essentially standardized here. So we could be comparing like to like, and we're getting a similar GMT, which should provide a similar level of efficacy. And again, despite the limited numbers, the efficacy that we're seeing matches the the sort of efficacy that we're seeing in this circumstance in adults, which in my mind, and we have others that can speak to this, is in the 70 to 80+% range.
Well, I would disagree with so I appreciate that, and thank you for running through that with me. I would disagree in some ways with the assumption that this is like to like in all contexts, because obviously it's an older age range, but it's also at a 30 microgram dose, which is 10 times the dose that we're studying in children. And so, I want to make sure not to make too many assumptions about the protection that a 3-dose primary series in kids 6 months to 5 years conveys. And again, whether a booster dose will be needed in this age range. Again, I feel comfortable with the fact that a 3-dose primary series has met the criteria for emergency use authorization, and I'm also anxious to get kids immunized and protected. But again, I feel like there are similarities in the 3 figures being shown here as you highlight, but there are differences. And I also want to acknowledge the differences, because I don't want to make too many assumptions about whether a booster dose will or will not be needed in kids 6 months through less than 5 years of age on that basis.
Yeah, maybe can I respond to that?
Please.
Oh, yeah. So let's face it. In the older adults, we're already talking about 4th doses that are being administered to high-risk populations. We're obviously contemplating what our future will be, informed by the VRBPAC meeting that's going to happen at the end of this month about what we do in the fall. So I think sort of discriminating, well, are the kids going to require a booster dose as opposed to the adults? I think everybody's going to require a booster dose, and it's just a matter of discerning with real-world effectiveness data and perhaps some of the additional control data when that booster dose is likely to occur. So I think for the purposes of this comparison, I just want to emphasize that this is after 3 doses in both groups at comparable sort of intervals of administration. We're getting the type of antibody that is likely to be associated with protection in the 70, 80+% range that we're seeing in adults, and the descriptive efficacy is matching that. So for this type of regimen, as a primary series in young kids, we're likely to see effectiveness that matches what we've seen in the clinical trial. I think, again, I would not bet against the vaccine here in this context.
Well, and just to be clear, I'm not betting against the vaccine. I'm just arguing for as much transparency about what we know and don't know with parents. And that includes sort of what to expect after a second dose and what to expect about whether additional doses will be needed. In that context, can you let us know about when you may have studies of safety and immunogenicity of a 4th dose in kids 6 months through 5 years of age? Thank you. Yeah,
so I think obviously that's under discussion, as well as what we decide to do, informed by the VRBPAC meeting at the end of this month. We already have plans afoot and are putting protocols together basically to look at moving forward with an Omicron vaccine or an Omicron plus a reference strain vaccine, or potentially other options that may be put forward in the setting of BA4 and 5. So we're going to tailor this to what seems to be best suited for what the future is most likely to bring in the fall. And so that work is already underway, and we're we are in and will be in further discussions with the FDA about that. But I think it does need to be informed by the decisions that are made at this month, so we don't go off on a tangent for something that, frankly, doesn't become the vaccine of the future.
Okay, thanks so much.
Thank you, Dr. Daly. I'm going to hand it over to Dr. Fink to respond, and then we're going to go with short questions for the last 3, and we have to move on to Dr. Oliver's presentation after that.
Thanks, Dr. Lee. While this slide is up, I just wanted to say a few things in the interest of full transparency. So first of all, this slide represents late-breaking data that Pfizer provided to us just shortly ahead of the VRBPAC, and we haven't had a chance to verify these immunogenicity analyses. Secondly, in the interest of comparing like to like, I just want to point out that a median 13-week interval between dose 2 and dose 3 is not an interval that has been evaluated, as far as I know, for efficacy. in adults. And so I would stress that we should be careful in trying to compare expected efficacy with this sort of dose 2 to dose 3 interval based on immunogenicity, looking at these pediatric age groups versus adults. Now, that being said, what we have seen time and time again with the data for these vaccines is that there is some level of efficacy after the first dose, and it increases after the second dose and increases again after the third dose. So it would not be unexpected that for this 3-dose primary series, the protection afforded by the third dose would be higher than the protection afforded after the second dose. We just don't have the confidence at this time, based on the small number of cases that are available to us, to cite a specific efficacy estimate. Thank you.
Thank you. Really quick questions, Dr. Paling.
Dr. Gruber, I wanted to ask you about febrile seizures. Did you have any febrile seizures, and what can you tell us about that in your study? Thank you.
Yeah, I'm happy to do that. Maybe, Charlie, if you can identify again the slide to bring up. We've got
Sure. Yeah, slide 43, 44, and 45. We can start with 43. Thank you.
Yeah, so let me talk first of all about febrile convulsions, because obviously in the setting of fever, and part of the reason we want to minimize fever with this vaccine series is to minimize this as a potential complication in those children that are predisposed. So we had 5 participants that had febrile convulsions. All were assessed as non-related to vaccine, and you can get some sense of why, because look at when they occurred with respect to vaccination. They were well distant from vaccination, whether we're talking about 6 months to less than 2-year-olds, or the 2 years to less than 5-year-olds, in association with other potential causes, except I guess for the last one that was down at the bottom, that was 21 days post-dose 1 in the vaccine group. And then if we go maybe to, would it be the next slide, Char, that talks about the
Correct. If we go to the next slide, 9. Mm-hmm.
Yeah. So if we go to the next slide, please. I'm not seeing it up yet on my screen. There we go. So for non-febrile convulsions, we had 6 participants had non-febrile convulsions. All were assessed as not related. One was a seizure, or what was believed to be a seizure, with eyes rolling back 2 days post-dose 2 in the 6-month to less than 2-year-olds in the vaccine group. Another seizure, 164 days post-dose 2, down at the 2 to less than 5, 147 days post-dose 2. In a case of status epilepticus, 18 days post-dose 1, someone known with a medical history of seizure disorder. So on balance, we think that, again, we're gratified that we've not seen, febrile convulsions that seem to be related to this vaccine, and nor do there seem to be related non-febrile convulsions. And I think, convinces us of, again, a satisfactory safety profile that I think is, again, enlarged by the fact that the level of fevers that we've seen has really been gratifyingly low.
Thank you. Dr. Sanchez, quick question.
Yes, I just wanted to know, were any of these mothers of these, especially of the younger children, vaccinated with, with COVID vaccine, or specifically with Pfizer, or was that an exclusion? And if they were vaccinated, how did you interpret antibodies baseline?
Yeah, so again, Char, you can remind us about the nature of the history that we obtained on these folks. I think one of the things to keep in mind, of course, is these children were starting to get vaccinated at 6 months of age. So the presumption is that any sort of passive antibody that they would have would likely have declined by the time that those children receive vaccine. I think, Dr. Sanchez, your question obviously becomes that much more important when we talk about children less than 6 months of age, and that'll clearly be an important part of the history. But Char, you want to just speak to the, what was discerned, if anything, about individuals that were vaccinated prior, and then I can make one other comment.
Sure, no, appreciate that. So we are collecting that data in terms of a maternal vaccination that's currently ongoing. We have not conducted a separate analysis, but as Dr. Gruber mentioned, it'll be part of what we look at. We don't have any other, we do have their baseline antibody testing for all children. They did have N binding at the time of entry into the study.
I will say something that will potentially be of interest, as I think maybe you know, we have a study 1015 in pregnant women who are vaccinated actually at the time of pregnancy, and we will be evaluating antibody in those children over time that are born. I think there, as you're aware, there is some information in the literature about protection afforded to those children by maternal immunization that looks promising, and that also actually impacts our thinking about, okay, well, what is the best course for protection of children less than 6 months of age? That figures into our discussions with regulatory authorities.
Thank you. I mean, it could go both ways also, as to whether there could be some interference if there is persistent maternal antibody. But thank you.
Thank you. And very last quick question from Dr. Long, and we do have to move on to Dr. Oliver's presentation and public comment, and we are running behind. So Dr. Long.
Yes, since the majority of children for whom we would potentially give this vaccine are already seropositive, I'm interested in the cadence of the response and the final antibody level in the 5-6%, I think, who were seropositive prior to immunization.
I don't know that I can, Dr. Long, I don't know that I can address the cadence because we didn't get serial sampling on these children. We actually have slides that show the antibody responses increased in those individuals that were seropositive, not surprising based on other experience we've had in other populations. But in the interest of time, maybe there's not a need to show that.
I'm okay with that answer. I just, when the primary infection for viruses sometimes is early in life, and these would have been in the first 6 months of life that they had their primary infection, or they still had antibody from maternal immunization now. Funny things can happen with immunization that they don't respond. So I'm happy enough to hear that they were at least as good as those that were seronegative.
Yeah. This is true for both the 6 month to 2 year olds as well as the 2 to 5. So it looks like even though those, as you said, some of those individuals by definition will have had to have been infected before 6 months of age to register, or they were PCR positive at the time that they actually were enrolled but asymptomatic. I think that's encouraging that in fact we still saw a good response above that actually seen in those that were naive.
Thank you.
Thank you. And thank you, Dr. Gruber, for patiently answering our questions today. We will move on to Dr. Sarah Oliver, who will be reviewing this information on mRNA COVID-19 vaccines in young children, providing a summary and workgroup interpretation.
Thank you so much, and good afternoon. Next slide. So I'll just briefly summarize the data we've seen so far today, a workgroup interpretation, and next steps. So first we'll talk about Moderna. Next slide. As a reminder, the trial was conducted from December of 2021 through of 2021 through February of 2022, with case accrual after a second dose at the height of the Omicron surge. Children were ages 6 months through 5 years of age in the US and were randomized 3-to-1 vaccine to saline placebo. The analyses were performed separately for 6 to 23 months and 2 to 5 years, and we have results pooled for a combined estimate for 6 months through 5 years. The Moderna schedule is 2 doses of 25 micrograms separated by 28 days, and the median follow-up time post-dose 2 is 2.5 months. Next slide. For the overall population, there were over 6,000 children included in the efficacy and safety analyses, again randomized 3-to-1. Then there was a subset of nearly 500 children included in the immunogenicity analysis. Next slide. For the efficacy endpoints among subjects with or without evidence of prior infection, for those 6 to 23 months, the VE was 50.9%, and for 2 to 5 years, the VE was 37%, for an overall VE of 41.5%. We have higher confidence in this estimate based on 181 COVID cases in the vaccine group and 97 cases in the placebo group. In addition, the efficacy in this trial is consistent with the post-authorization vaccine effectiveness for the Moderna vaccine in adults 18 through 64 years during Omicron, where the VE against infection 2 months after dose 2 was 35%. Next slide. Then regarding immunogenicity data, the antibody levels were measured 28 days after the second dose for participants without prior infection. The antibody responses after 2 25 microgram doses in children ages 6 months through 5 years compared to 2 100 microgram doses in individuals aged 18 to 25 years met the non-inferiority criteria with the ratios shown here. Next slide. Then regarding safety data, no deaths were reported in any trial participants. Serious adverse events were rare overall, occurring in 0.5% of vaccine recipients and 0.2% of placebo recipients. One vaccine recipient had 2 SAEs, a fever and febrile seizure, that was deemed possibly related to the vaccine by FDA. No cases of myocarditis or anaphylaxis were noted in any trial participants. Next slide. Local reactions, such as pain at the injection site, were common, and systemic reactions also occurred. In the children ages 2 through 5 years, fatigue and headache were the most common, and in the younger children 6 through 23 months, irritability and sleepiness were most common. Symptoms typically started 1 to 2 days post-vaccine and resolved after 2 to 3 days. Next slide. Fevers were more common after vaccine than placebo, and more common after dose 2 than dose 1. Most fevers were reported in the first few days after vaccine and lasted for around a day. The table at the bottom shows fevers after the second dose at 16%. However, fevers for other routine vaccines given at this age, such as the pneumococcal vaccine, can be around 30%. Then, as we heard from Moderna, there was 1 febrile seizure that was possibly related to the vaccine. Next slide. The workgroup also noted imbalances with some respiratory infections, such as RSV. However, when taken for all respiratory infections, including COVID, no imbalances were noted. The events were rare in less than 1% of trial participants. In addition, there was no patterning or clustering of the cases, and the clinical consistence were typical and consistent with the seasonal respiratory infections. In addition, testing was not performed systematically for all patients and was at the discretion of the clinician evaluating the patient. Because of this, testing for additional respiratory pathogens varied and may have varied by the results of COVID-19 testing. Then for the Moderna trial, lymphadenopathy was a solicited adverse event. So lymphadenopathy, either in the axillary region or the groin, was noted in 9% of vaccine recipients compared to 2% of placebo recipients. Next slide. So in conclusion, the efficacy seen after 2 doses of the Moderna COVID-19 vaccine in children ages 6 months through 5 years of age is consistent with real-world vaccine effectiveness in other ages during Omicron predominance. The antibody levels after 2 doses in children ages 6 months through 5 years produces similar antibody levels seen after 2 doses in individuals aged 18 through 24 years. And the reactogenicity post-vaccine is consistent with other recommended vaccines in this age group. Next slide. Moving to Pfizer. Next slide. So to describe the clinical trial, the trial was conducted from June of 2021 through April of 2022. Children ages 6 months through 4 years in the US were randomized 2-to-1 vaccine to a saline placebo. As always, as we heard before, the analyses were performed separately for the 2 ages and then pooled for a combined analysis. This series is 3 doses of 3 micrograms each. Dose 1 and dose 2 were separated by 21 days. Then dose 2 and dose 3 are separated by at least 8 weeks. However, note the interval between dose 2 and dose 3 in the trial is longer than the authorized interval. Among children 6 to 23 months, the interval was 16 weeks with a range of 8 to 32 weeks. And for children ages 2 to 4 years, the interval was 11 weeks with a range of 8 to 34 weeks. In addition, the median follow-up time post-dose 3 was 1.3 months. Next slide. Then I just want to walk through some of the timing with the Pfizer trial. For children 2 to 4 years, they began giving the first doses in June and second doses starting in July. However, per protocol, they began unblinding participants 6 months after the second dose, which was around December of 2021 and into January and February. Then, as we're aware, based on analyses after the second dose, the protocol was amended in February and third doses were given. However, by then, many of the initial cohort from the trial were unblinded and placebo crossover had already occurred. After FDA's request to expand the size of the trial, they began an expanded enrollment, initially extending for an expanded safety enrollment. This cohort began receiving doses in October and November, with, again, third doses starting in February and March. But if we look at the bottom, you can see we have blinded person time contributing to the dose 3 efficacy and safety evaluation from February through April, which was when the cases began declining after the Omicron surge. Next slide. This slide walks through everything again with the 6 through 23 month population. The same phenomenon occurred where participants were unblinded 6 months after the second dose. Next slide. So if we walk through the impact that had on the total number of participants who contributed blinded person time to the efficacy and safety evaluations, on the left in the 2 to 4 years, you can see they started with 2,700 children in the 2 to 4 year group, and then on the right, in the 6 to 23 month group, started with around 1,700 children. By dose 3, much of that population had been unblinded, and overall, 32% of the overall eligible population contributed blinded person time to the efficacy and safety evaluations. Next slide. So then this slide shows the efficacy estimates after the third dose. For those 6 to 23 months, 75.5%, and for 2 to 4 years, 82.3%. Overall, in the 6 months through 4 years, 80.3%, but note wide confidence intervals for all. We have lower confidence in this estimate as these were based on 3 cases in the vaccine group and 7 cases in the placebo group. For comparison and context, the post-authorization vaccine effectiveness for the Pfizer COVID-19 vaccine in adolescents aged 12 to 15 years during Omicron, the VE against infection 2 months after dose 2 was 29%, and 2 months after dose 3 was 43%. Next slide. Overall, the workgroup felt that the post-dose 3 efficacy data are difficult to interpret. There is a limited number of cases during the blinded follow-up. In addition, the protocol specified a need for 21 cases prior to the formal efficacy analysis, and only 10 were included in the interim descriptive analysis. Importantly, the dosing interval between dose 2 and dose 3 varied and are longer than the authorized interval. And the median blinded follow-up time is limited, with a median time of 35 to 40 days for the age groups here. Next slide. Again, the antibody levels were measured 1 month post-dose 3 for participants without prior infection. Antibody responses after 3 microgram doses in children 6 months through 4 years were compared to 2 30 microgram doses in individuals aged 16 to 25 years. Both age groups met the non-inferiority criteria with the ratios shown here, and the numbers included in the population here were 225 children total. Next slide. Then for comparison and context, the workgroup again reviewed the results that were seen after dose 2. For 6 months to 23 months, minimal VE was seen, but the non-inferiority criteria were met. For 2 to 4 years, some protection was noted with a VE of 33%, but the non-inferiority immunobridging criteria were not met. Next slide. From a safety standpoint, No deaths were reported in any trial participants. Again, serious adverse events, or SAEs, were rare overall and occurred in 1% of vaccine recipients and 1.5% of placebo recipients. One vaccine recipient had 2 SAEs, fever and pain in an extremity requiring hospitalization, possibly related to the vaccine, but the FDA felt symptoms were potentially consistent with a viral myositis. And again, no cases of myocarditis or anaphylaxis were noted in trial participants. Next slide. Local reactions such as pain or tenderness at the injection site occurred within 7 days and were common. Fatigue was the most common systemic symptom in children 2 to 4 years. Irritability and drowsiness were more common in children ages 6 through 23 months. Overall, the reactions were comparable after dose 1, 2, and 3. Most symptoms were mild and resolved after 1 to 2 days. Next slide. Then specifically for fevers were reported with similar frequency after both vaccine and placebo, and similar frequencies after doses 1, 2, and 3. Frequency after the second and third doses are shown here. Then most fevers were reported on days 1 and 2 after any dose and lasted for a median of 1 day. Next slide. So in conclusion, antibody levels after the 3 doses in children ages 6 months through 4 years produced similar antibody levels after 2 doses in individuals aged 16 to 24 years. Reactogenicity post-vaccine was similar after each of the 3 vaccine doses and similar to reactions seen in placebo recipients. However, the efficacy estimates after 3 doses are difficult to interpret given the small numbers and limited follow-up time, and the workgroup noted that the impact of the longer interval in the trial between dose 2 and dose 3 on efficacy, reactogenicity, and safety are unknown. Next slide. So the workgroup interpretation in total, next slide. The mRNA COVID-19 vaccine clinical trials in young children were both conducted during Omicron predominance, but at different months and incidence levels. So in addition to differences in the number of participants in the efficacy analyses and differences in follow-up time, these incidence levels impacted COVID-19 case accrual and certainty in the efficacy estimates. Because of this, the efficacy estimates for these 2 mRNA vaccines cannot be directly compared. However, both vaccines met the non-inferiority criteria for neutralizing antibody levels. Next slide. Note that the current data are for either a 2-dose or a 3-dose primary series. To achieve the criteria set by FDA for authorization, 2 doses for Moderna or 3 doses of the Pfizer vaccine were required. For ages 5 and over, 2 doses achieved the preset efficacy requirements or the required antibody levels for immunobridging. A booster was then provided to optimize the immune response and address waning of antibody titers detected after completion of the primary series. Post-authorization effectiveness studies can help determine the subsequent timing and need for boosters after a 2-dose with Moderna or 3-dose with Pfizer primary series. That booster would then be a 3rd dose for the Moderna vaccine and a 4th dose for Pfizer in this age group. Next slide. However, we do know that in other age groups during Omicron, the mRNA COVID-19 vaccine post-authorization vaccine effectiveness was lower against infection, but higher protection was noted against severe disease, as we heard from Dr. Link-Ellis earlier today. The clinical trials were not powered to detect efficacy against severe disease in this young population, but similar patterns in this age group are expected to what we see in everyone ages 5 and over. Next slide. Then for next steps, the evidence to recommendation framework, including the grade summary of the data, will be presented tomorrow. For ETR tomorrow, we will be discussing 2 separate policy questions. So should vaccination with the Moderna vaccine be recommended for children 6 months through 5 years of age under an EUA? And should vaccination with the Pfizer vaccine be recommended for children 6 months through 4 years of age under an EUA? We will not be comparing the 2 vaccines. We'll be comparing each vaccine to no vaccine. Next slide. just want to acknowledge our team that helped pull all of these slides and a careful review of the data. Next slide. That's it. I'll turn it back over to you, Dr. Lee. Thanks.
Thank you, Dr. Oliver. This presentation is now open for questions. Dr. Lair.
Thank you, Dr. Oliver, again for a lovely presentation, very informative. I want to see if you can convey the nuances the work group felt about the Pfizer vaccine and the difficulty in evaluating the efficacy compared to it meeting the FDA criteria, and if that was enough to give the work group pause about approval for the Pfizer vaccine.
Thanks. I'm happy to maybe kind of start that discussion and then we can finish it tomorrow, because I do think some of this may come out in some of the ETR determinations as we walk through everything. I will note, the work group again discussed that with the 2 doses of Moderna or with 3 doses of Pfizer, it met the criteria set out by FDA to grant the EUA. So I think the work group was reassured in that, but there was considerable discussion about the differences both in the follow-up time and the fact that there was limited kind of person time, both by number of participants and by time after that 3rd dose with Pfizer, and considerable discussion that we'll need to monitor post authorization safety and effectiveness very closely with both vaccines, as we always do, but that it'll be especially important with Pfizer as we're watching what the efficacy ends up being, especially given the fact that it is likely that many children or most children will receive the vaccine at that 2nd to 3rd dose at an 8-week interval, and that the data to inform what that effectiveness looks like is actually pretty limited based on the trial. And I don't know if Dr. Daly wants to comment anything else, or if he thought that was.
Actually, I think we're limited on time, so why don't we wait until tomorrow to
Okay.
address some of these other issues.
Thank you. Dr. Long?
I don't know if you want to wait till tomorrow to discuss this, but I think we should discuss what you've just been talking about. And I decided when I first saw these data, especially about Pfizer's efficacy data, that I think we should just assume we don't have efficacy data. And I think that I would be comfortable enough with the immunobridging. And that I think should be something that we should discuss, but we certainly have done other vaccines where we have not had enough time or the capability of efficacy that we've used immunobridging. So although we don't have that magic number, we do have the data that if you think that the antibody tests as they're performed by different pharmaceutical groups are measuring the same antibody and are comparable with the ones that they have done in other age groups, I'm not uncomfortable with considering that we don't have efficacy data.
Dr. Fink, did you want to respond?
I just wanted to echo what Dr. Long said, that from FDA's perspective, we would have been confident in the clinical benefit of this 3-dose primary series based on the immunobridging data alone, even if there were no efficacy data available. And where we're less confident is quoting a specific efficacy estimate. Thank you.
Thank you. Dr. Daly, did you want to add anything?
Oh, just that from the COVID vaccines workgroup point of view, our discussion was very much along the lines of what Dr. Long said, and our interpretation was along the lines of what Dr. Long and Dr. Fink said. Thank you.
Are there any additional questions or comments for Dr. Oliver? And I don't want to stifle discussion, but I do want to make sure that I want to make sure we have enough time for that discussion because I think it's incredibly important for our consideration of the votes. But we do have an additional day tomorrow for this discussion with additional context from presentations. Any other questions from our ACIP members or our liaisons? Okay, I don't see any additional hands raised at the moment. What I'd like to do is ask if our colleagues in the room at CDC can prepare us for a public comment section. I'd like to welcome and thank our public comment speakers for addressing the committee today. All of the speakers today submitted a request in advance of the meeting, and the final list of public commenters was determined via a lottery. For our speakers today, we have a limited public comment period, and apologies for starting us late. And in order to make it through all of the listed speakers, it's extremely important that each speaker limits his, her, or their remarks to 3 minutes. We will be displaying a timer on the screen so you know how much time you have left. As a reminder, ACIP is very interested in hearing public comments about the agenda topics being discussed today. Our committee appreciates diverse viewpoints that are respectful in nature and issue focused. We do ask speakers to share their views about the issues being discussed rather than comments about individuals. And I want to thank our speakers again, and we look forward to your comments. So the first public comment speaker I have is Daniel Rocker.
Good afternoon. My name is Daniel Rocker, and I'm speaking today on behalf of Retire Safe, a grassroots nonprofit organization formed in 1991. Retire Safe's mission is to educate and advocate on behalf of mature Americans on issues including Social Security, Medicare, health, safe retirement, and financial well-being. We're grateful to have the opportunity to present our comments today. As we've said many times before in both oral and written comments of this committee, the importance of vaccines cannot be overstated. All of us, from the youngest to the oldest, are at risk from COVID-19. We know the devastation it has caused on older Americans, as over 75% of deaths have been among adults 65 and over.
Apologies. I hate to interrupt. I'm so sorry, but the timer is not working on the screen. So I just want to ask my CDC colleagues if they could start that.
Do I restart or continue?
No, please go ahead and continue. Thank you.
Okay, thank you. Nonetheless, seniors were among the first group to become eligible for vaccination and took full advantage of it. 95% of those over the age of 65 have received at least one shot, and over 91% are fully vaccinated. But for many seniors, concern, even fear, was not so much about themselves, but for their families, especially their grandchildren and their great-grandchildren, who have had no access to protection. Yesterday, that long wait ended with the FDA's authorization. Now we look to you to give recommendations that will enable providers to begin to protect our smallest loved ones with these life-saving vaccines. We thank you for the swiftness in which you have met today, only one day after FDA. Retire Safe would like to ask one more thing. The threat of COVID has been and remains unprecedented, especially to older adults. But it's not the only danger to older Americans' respiratory health. Last year, the FDA approved 2 pneumonia vaccines, which are of enormous value to seniors, millions of whom are still waiting for this committee to issue recommendations for their situation. We're so thankful for ACIP's immediate turnaround time for COVID for young children. We ask you to please act swiftly now on behalf regarding pneumonia disease. Thank you.
Thank you very much for your comment. We will move to the next public comment speaker, Mrs. Kate Schenk.
Good afternoon. Thank you for allowing me the opportunity to speak today. My name is Kate Schenk. I'm the mother of 3 children under the age of 5. The last 2.5 years have been long and challenging for families like mine who have young children. My husband and I have done everything we can to protect our children from COVID-19. Our children haven't been inside grocery stores or department stores, let alone typical childhood staples, such as indoor amusement parks, museums, malls, and restaurants. A safe and effective vaccine for children under 5 will allow our children to experience the joys of playdates, vacations, family gatherings, and so much more. My oldest child is incredibly excited to be starting kindergarten this September, but she doesn't turn 5 until mid-August. Moderna's under 5 vaccine will allow her to start the school year fully vaccinated. Both Moderna and Pfizer are safe and effective vaccines. They have met Immunobridging with similar efficacy to adult vaccines during the Omicron surge. Their safety profiles are excellent, both in the pediatric trials as well as in the millions of doses that have been given to older cohorts. These vaccines can protect against the most severe of outcomes, the things that have worried parents since the beginning of the pandemic, death and hospitalization, and can potentially decrease the risk of other complications, including long COVID, MIS-C, neurological effects, diabetes, and hepatitis. At this point in the pandemic, many people have seemingly moved on and are completely forgoing COVID mitigation efforts, such as masking and social distancing, and our youngest children have been left behind, unprotected. Those without children do not even recognize the toll this has taken on young families, since we have become invisible while they are living life as normal. Vaccination can change this. Please recommend COVID-19 vaccination for all children under the age of 5. We know that COVID-19 affects children. In April, it was estimated that 3 out of 4 children have had COVID. Millions of children have been infected. 4 times more children have been hospitalized this year with the Omicron variant than with Delta last year, and the largest increase was in children too young to be vaccinated. 482 children under age 5 have died. In addition to recommending vaccination for all children under age 5, please ensure that all children have easy access to the vaccine. Low-income families, families who rely on public transportation, and families without an established pediatrician need to be given the opportunity to vaccinate their children promptly. Anything that can be done to provide easy and equal access to vaccination should be prioritized. After all, we've been waiting so long for this. Finally, the delays we have seen in the under 5 vaccine have been frustrating and have undoubtedly led to illness, hospitalization, and death during the wait. In the future, for pediatric boosters and other vaccines, please ensure greater prioritization and expeditious review is given so our children are not left behind yet again. I urge you to please approve these vaccines today so parents will have the ability to protect their children from COVID-19. Thank you for your time.
Thank you for your comment. Next, we have Mrs. Caitlin Combe.
Good afternoon. My name is Caitlin Combe. Thank you so much for the opportunity to speak today. I am a 2nd grade special education teacher and the mother of a pandemic baby, a determined, joyful, spunky 2.5-year-old. I have no conflicts. I am here today to advocate for vaccine access for children under 5. I know that you are well versed in the data of this pandemic, so I would like to take a moment to speak to the personal side. My daughter has lived all but the first 3 months of her life in a pandemic. We have followed all of the CDC guidelines throughout, which for her means that she did not get to have a first birthday party. We have missed out on celebrating several holidays with family. She has never been to indoor spaces like museums, and she has missed out on countless other activities that are normally considered typical for a child her age. She lives vicariously through books, and it is heartbreaking to hear her say, "Mama, someday I will go to an aquarium like Curious George." Or, "Mama, sometime we will go to a grocery store like Pete the Cat." These are things that should be taken for granted, not things that have a mythical connotation. It is also heartbreaking to watch her give her stuffed animals a COVID test during her pretend play, and then turn to me to say, "Don't worry, Mama, the COVID test is negative." Each day, I have to weigh the risk of every activity, as all mitigation measures have been dropped and there are no protections for my daughter, other than the ones I can cobble into place. Each day, I put a very small mask onto her little tiny face and hope that it will be enough to keep her safe while she is at daycare, while knowing that she must spend several hours unmasked while she eats 2 snacks and a lunch and while she naps. Each day, I check the wastewater levels in my section of the state to see what direction they are trending in. I try to complete some complicated mental calculus to figure out how much risk I'm exposing her to on a daily basis, between her time in daycare, my exposure as an elementary special education teacher, etc., and I'm exhausted. So far, we have been lucky and are part of the very small percentage of people left in this country who have not been infected by COVID. I am concerned about the unknown long-term ramifications of COVID infection, especially for someone who is unvaccinated like my child. A vaccine, while not a magic bullet, would certainly change all of this for us. Moderna and Pfizer have both demonstrated that their vaccines are safe and effective. Both met the Immunobridging standards and both protect against severe illness, hospitalization, and death. Both vaccines demonstrated similar efficacy to the adult dose during the Omicron surge. Children under 5 deserve to receive the protection that these vaccines provide, just like everyone else in the country, and their parents deserve to be able to make a choice between the two vaccines. These delays that have persisted throughout this process for children under 5 cannot be allowed to happen again. Many children have become sick, have been hospitalized, and have even died while waiting for approval of these vaccines. I cannot urge you strongly enough to approve these vaccines today for children under 5 to provide parents with the ability to protect their children. Thank you again so much for giving me the time to speak today. I appreciate it.
Thank you for your comment. Next, we have Dr. Andrea Klein-Tilford.
Greetings. I am Andrea Klein-Tilford, and I do not have any conflicts to report. I'm a pediatric nurse practitioner and the president of the National Association of Pediatric Nurse Practitioners, a professional organization representing more than 8,000 pediatric-focused nurse practitioners. We support the timely and complete immunization of all eligible infants, children, and adults to maximize the health and well-being of all people. In the United States, children have accounted for roughly 19% of all reported COVID-19 cases. While some children experience few or no symptoms, some children with COVID-19 develop long-term complications, severe illness such as multisystem inflammatory syndrome, require hospitalization, or worse, die from COVID-19. According to the CDC, hospitalization rates of children under 5 during the Omicron surge were 5 times the rate during the previous Delta variant. It's critical that children of all ages have access to safe vaccines as soon as possible to prevent disease and hospitalization as COVID continues to spread and mutate and before we enter the traditional flu and RSV season. Experts agree that the first 5 years of a child's life are especially crucial regarding physical, intellectual, and social emotional development. During these early years, the brain grows faster than at any other stage in life, with many in this age group being too young to adhere to the mitigation strategies such as consistent physical distancing and mask wearing. Your approval of COVID-19 vaccines for children under 5 would significantly help protect this age group and allow many families to return to a sense of normalcy. COVID vaccines have the potential to help young children enjoy typical age-appropriate socialization and achieve developmental milestones that are critical for their long-term physical and mental health. For months, parents and caregivers of young children have been desperate for a safe, effective vaccine. Based on the available safety and efficacy data, NAPNAP urges ACIP to approve administration of the Pfizer-BioNTech 3-dose vaccine for children 6 months through 4 years and the Moderna 2-dose vaccine for children 6 months to 5 years, based on the FDA's emergency use authorization recommendation. We also call on the FDA and CDC to work with manufacturers and state and local agencies to provide comprehensive, equitable vaccine delivery to every eligible child in the United States as quickly as possible. Being able to vaccinate all people 6 months and older means we'll finally have the necessary tools to fight this ongoing pandemic and improve our nation's public health. Thank you for this time to share the views of the National Association of Pediatric Nurse Practitioners and my own views as a mother of 2 children under 12 years of age.
Thank you for your comment. Next, we have Mr. Christopher Royster. Mr. Royster, I believe you are still muted.
Yes, can you hear me?
We can, thank you.
Thank you. I was having problems with my audio. Good afternoon. My name is Christopher Royster. Thank you for allowing me to present a public comment today. As I am a representative for Retire Safe, as mentioned by my colleague, Mr. Daniel Rocker. I believe that these meetings are very important and paramount for all age groups. Many have perished during this COVID-19 pandemic. However, the current state of our society is filled with adults, including the elderly.
Mr. Royster, could you pause the timer, please? Thank you. I think we've lost you, and we had a little bit of an echo, too. Are you able to speak?
Hello?
There you are. Please go ahead. We'll add 20 seconds to your clock. Please go ahead.
Thank you. However, the current state of our society is filled with adults who are currently vaccinated, the same as the elderly. Thus, to protect the lives of our senior citizens, it's very important that you all pass these vaccination laws and make it a requirement for all of our youth. From listening to the presentations today, I believe that a 3-dose Moderna vaccination criteria is necessary for all age groups. After listening to the results and details of each presentation, I believe that Moderna has been proven to be an efficient vaccine thus far. With the limited time period for which the development of these vaccines have been subject to, and the 70% to 80% efficacy results for a 3-dose, which has been presented today, I believe that that is enough for me to believe that these vaccinations should be used and should be implemented for all children. Our youth are the future. Therefore, we must protect our children as they are the catalysts which will move our society forward. We must protect them at all costs. Thank you.
Thank you very much for your comment. Next, we have Amy Baker. Amy Baker, are you able to unmute your line?
She was on, Dr. Lee, but maybe she lost her connection.
Okay, thank you. Why don't we move to Evelyn Pinheiro?
Good afternoon. Can I start? I'm a parent to a 4.5-year-old and a 1-year-old, and I want to thank you for meeting so quickly, even on Father's Day weekend. Like you, I know that every day counts if we want our children to be vaccinated before infected. Every day, more people take off their masks at my son's school. Every day, I'm notified of new COVID cases there. And every day, my unvaccinated children grow older without being able to travel to see grandparents and cousins. Previous speakers have already talked about hospitalization and death among children under 5 with COVID. One of the other risks that keeps parents up at night is the potential for chronic problems. Today is my 1-year-old's first birthday, and he has his entire life ahead of him. We don't know the long-term effects that this novel disease might have on the newly developing brain or lungs or heart. And we do not want to take the risk for our children of long COVID, which we know vaccines can decrease. So like thousands of parents, we continue to live like it's March 2020. While it takes a village to raise children, we are doing it alone, and we're so tired. We miss getting to share the arrival of our babies with family and friends. We miss library story hours and indoor play dates. My older son, an avid dinosaur lover, is dying to get his COVID shot so he can go to the Natural History Museum. I want to be able to more safely visit with elderly loved ones. June 15th was like Christmas morning to the many 6,500 parents and doctors who belong to a group called Protect Their Future, formed to advocate for vaccines for under 5s. I implore you today to approve both vaccines as they were both safe and met the primary endpoints of immunobridging. But once these vaccines are approved, we also need help accessing them. My peers in Florida are frantic as they live in the only state that didn't order vaccines. Here in New York, where I live, state sites are closing, and my county isn't offering the shot to under 5s. Pharmacies cannot vaccinate children under 18 months. Some pediatricians aren't offering the shot. For most of us, Moderna will be the only option to fully vaccinate our children before the beginning of the school year. Even though I live in a New York City suburb, the closest place I can find that will be giving Moderna to children under 18 months is 3 hours away. In addition to approving both vaccines for children under 5, please help give parents a choice of vaccines and more options for vaccinating our babies.
Thank you for your comments. Next, I'll ask if Amy Baker is on.
Yes, I was on. Sorry, I was trying to unmute with my phone and not star 6.
That's okay. Please go ahead.
Thank you for giving me the chance to speak today. I can go through the facts, such as it's been 875 days since the first COVID case was reported in my state, and 481 children under 5 in the U.S. have died from COVID. But I'll stop there because I hope you know these facts given your position. Instead, I'll explain what our lives have looked like the past 27 months trying to keep our 1- and 3-year-olds safe from COVID. No daycare, quitting my job because no safe daycare, and it becomes impossible to balance a job and caring for my children once my spouse returns to the office. No first, second, or third birthday parties. Missing weddings, missing funerals, no play dates or library story times, no peer social interaction for our children, no reprieve for us parents, day in, day out, for 826 days. 231 days since children 5 and up could get vaccinated, and we thought it'd be a matter of weeks until it was our turn. 129 days since it was announced that Pfizer would be submitting for under 5s on a rolling basis at the FDA's request, and we thought it'd be any day. At a minimum, that's 129 days that pediatricians, county health departments, and medical offices have had to prepare and plan for the hopefully eventual administering of the COVID vaccine to children under 5. 129 days that someone in overseeing bodies had to write up guidelines for the rollout of pediatric vaccines once they received all the proper approvals. And that hasn't seemed to happen, though, because after we've sacrificed and after we've waited and after we fought for and advocated for our children, we are still fighting to protect our kids. We can pre-order books and phones, but we can't find any answers about where we'll find a vaccine. The county says to contact your pediatrician. The pediatrician says to try the county or the medical group. The medical group says they won't offer vaccines and contact the pediatrician. Some places don't know when they'll have it or how it'll be administered, nor will they confirm if they pre-ordered, but yet they will say that they'll only have the vaccine that will take an extra 7 weeks to get through the primary series. Or the parents in Florida who want to protect their kids with vaccination are stuck in a state that has refused to pre-order the vaccine, and the privileged ones are stuck driving hours to get it. I've seen countless parents continue to feel defeated by the lack of communication, and after the months we've waited, it's unacceptable. The bottom line is this: both of these vaccines met their immunobridging and safety endpoints. Therefore, if we are still recommending this vaccine for all children over 5, today we need to be recommending it for all children under 5. COVID can result in serious harm to our children, and the messaging from our governmental agencies that children are not vulnerable to these harms needs to change. We've seen the vaccine is safe. Other countries, such as Germany, have been allowing its off-label use for months, and we've seen the results of rigorous trials that thousands of children have bravely been a part of. And while we know that additional boosters will be required and that this is not the end of the road, it is our only hope at this point to taking a step towards normalcy when we live in a country that has abandoned all protective measures and left these kids behind. Thank you.
Thank you for your comment. I want to thank all of our public comment speakers today. We really do appreciate everyone who contributes to this meeting through both written and oral public comment. And with that, that is our last agenda item for today. So I would like to thank all of our speakers and our members and liaisons for the conversation today. We look forward to meeting again tomorrow for additional presentations and for votes on these 2 vaccines. Dr. Wharton, are there any further announcements?
No, we will be reconvening tomorrow morning at 10 a.m. Eastern Daylight Time.
Thank you. So today's ACIP meeting is now adjourned, and we look forward to talking to everyone tomorrow. Thank you.